Chapter 8, Recombinant DNA Technology. Subtopic 8.2 Methods in Gene Cloning. Step 1, Isolation of gene. That is, isolate plasmid from bacterial cells such as E. coli. Meanwhile, target DNA containing gene of interest isolate from any organism of interest. The key word here is isolate, target DNA, plasmid. Step 2, Cleave or cut. The plasmid and DNA that isolated are cut by same restriction enzyme at restriction site so that it produce restriction fragment which is complementary to the end of the plasmid that had been cut. The key word in this step is same restriction enzyme. Step 3 is insertion. This means insertion of restriction fragment into plasmid. For example, sticky of plasmid join with sticky end of restriction fragment. DNA ligase used to join covalently both DNA molecules forming recombinant DNA. The key word is DNA ligase. Step 4 is transformation and amplification. Transformation is the transfer of recombinant DNA into bacterium host cell. For example, ligation product is put into bacteria and treat the bacteria with heat and presence of CaCl2. The recombinant DNA will transfer into host cell. Then, the bacteria is grown or reproduced in a culture to form clone cell. The key word is transfer, host cell, heat, and CaCl2. Step 5 is screening. Screening is the step to obtain the desired cloned bacterial cells carrying the gene of interest. Let the bacteria reproduce on medium containing ampicillin and sugar called X-cal. Ampicillin ensures that only bacteria that contain plasmids will grow. Because plasmid have AMPAR genes that produce protein that protect bacteria from ampicillin. The key word is contain plasmid will grow, AMPAR protect bacteria. By using X-cal, cloned bacteria with non-recombinant plasmid will appear as blue color because it have intact LAC-Z, that is gene codes for beta-galactosidase which hydrolyze X-cal to yield a blue product. So white colonies shows bacterial clones which carry recombinant DNA plasmid. The key word is, only colonies that alive and show white colonies when reproduce in ampicillin and X-cal. For more easier to remember the step of gene cloning, you all can remember the sentences. I cut insect and Nolly shouts. Thanks for watching.